Next is about membrane-bound antigen receptor complexes. The recognition of antigen by both B and T lymphocytes occurs at the cell surface. An antigen receptor on a B cell receptor or BCR, consists of a membrane-bound immunoglobulin that binds selectively to a portion of an intact antigen. That's an epitope. So, in contrast, the antigen receptor on a T cell receptor or a TCR, recognizes and binds to a small fragment of an antigen, typically a peptide about 7 to 25 amino acids in length, that is held at the surface of another cell. Both types of antigen receptors are part of large membrane-bound protein complexes that include invariant proteins. So the invariant polypeptides associated with BCRs and TCRs play a key role in transmitting signals to the interior that lead to changes in activity of the B cell or T cell. Each subunit of a TCR contains two Ig-like domains, indicating that they share a common ancestry with BCRs. So, like the heavy and light chains of the immunoglobulins, one of the Ig-like domains of each subunit of a TCR has a variable amino acid sequence, the other domain has a constant amino acid sequence. X-ray crystallographic studies have shown that two types of antigen receptors also share a similar three-dimensional shape. Next topic is about the major histocompatibility complex. During the first part of the 20th century, clinical researchers discovered that blood could be transfused from one person to another as long as the two individuals were compatible for the ABO blood group system. The success of blood transfusion led to the proposal that skin might also be grafted between individuals. This idea was tested using World War II when skin grafts were attempted on pilots and other military personnel who had received serious burns. The grafts were rapidly and completely rejected. After the war, researchers set out to determine the basis for tissue rejection. It was discovered that skin could be grafted successfully between mice of the same inbreed strain, but that grafts between mice of different strains were rapidly rejected. So, mice of the same inbred strain are like identical twins, they are genetically identical. So, subsequent studies revealed that the genes that govern in tissue graft rejection were clustered in a region of the genome that was named the major histocompatibility complex or MHC. Approximately 20 different MHC genes have been characterized, most of which are highly polymorphic. Over 7,000 different alleles of MHC HLA genes have been identified, far more than any other loci in the human genome. It is very unlikely, therefore, that two individuals in a population have the same combination of MHC alleles. This is the reason that transplanted organs are so likely to be rejected and why transplant patients are given drugs such as cyclosporin A to suppress the immune system following surgery. So, cyclosporin A is a cyclic peptide produced by a soil fungus. So, cyclosporin A inhibits a particular phosphatase in the signaling pathway, leading to the production of cytokines required for T cell activation. Although these drugs help prevent graft rejection, they make patients susceptible to opportunistic infections similar to those that strike people with immunodeficiency diseases such as AIDS. It is obvious that proteins encoded by the MHC did not evolve to prevent indiscriminate organ transplantation, which raises the question of their normal role. Long after their discovery as transplantation antigens, MHC proteins were shown to be involved in antigen presentation. Some of the key experiments that led to our current understanding of antigen presentation are discussed later. So, it was noted earlier that T cells are activated by an antigen that has been dissected. So cyclosporin A is a cyclic peptide produced by a soil fungus. Cyclosporin A inhibits a particular phosphatase in the signaling pathway, leading to the production of cytokines required for T cell activation. Although these drugs help prevent graft rejection, they make patients susceptible to opportunistic infections similar to those that strike people with immunodeficiency diseases such as AIDS. So it is obvious that proteins encoded by the MHC did not evolve to prevent a discriminate organ transplantation, which raises the question of their normal role. So long after their discovery as transplantation antigens, MHC proteins were shown to be involved in antigen presentation. So some of the key experiments that led to our current understanding of antigen presentation are discussed later. So it was noted that T cells are activated by an antigen that has been dissected into small peptides and displayed on the surface of an antigen presenting cell. These small fragments of antigen are held at the surface of the APC in the grip of MHC proteins. Each species of MHC molecule can bind a large number of different peptides that share certain structural features that allow them to fit into its binding site. For example, all of the peptides that are capable of binding to a protein encoded by a particular MHC allele, such as HLA-B8, may contain a specific amino acid at a certain position, which allows it to fit into the peptide binding group. Given the fact that each individual expresses a number of different MHC proteins, and each MHC variant may be able to bind large numbers of different peptides, a dendritic cell or macrophage should be able to display a vast array of peptides. At the same time, not every person is capable of presenting every possible peptide in an effective manner, which is thought to be a major factor in determining differences in susceptibility in a population to different infectious diseases, including AIDS. For example, the HLA-B35 allele is associated with rapid progression to full-blown AIDS, and the HLA-DRB1 1302 allele is correlated with the resistance to a certain type of malaria and hepatitis B infection. The MHC alleles present in a given population have been shaped by natural selection. Those persons who possess MHC alleles that are best able to present the peptides of a particular infection agent will be the most likely to survive an infection by that agent. So conversely, persons who lack these alleles are more likely to die without passing on their alleles to offspring. As a result, populations tend to be more resistant to diseases to which their ancestors have been routinely exposed. This could explain why Native American populations have been devastated by certain diseases such as measles, which produce only mild symptoms in persons of European ancestry. The entire process of T cell mediated immunity rests on the basis that small peptides derived from the proteins of a pathogen differ in structure from those derived from the proteins of the host. So consequently, one or more peptides held at the surface of an APC serve as a small representation of the pathogen, providing the cells of the immune system with a glimpse of the type of pathogen that is hidden within the cytoplasm of the infected cell. So nearly any cell in the body can function as an APC. So most cells present antigens as an incidental activity that alters the immune system to the presence of a pathogen, but some professional APCs, dendritic cells, macrophages, and B cells, are specialized for this function. So, when a T cell interacts with an APC, it does so by having its TCRs dock onto the MHC molecules projecting from the APC surface. This interaction brings a TCR of a T cell into an orientation that allows it to recognize a specific peptide displayed within a groove of an MHC molecule. The interaction between MHC proteins and TCRs is strengthened by additional contacts that form between cell surface components such as occur between CD4 or CD8 molecules on the T cell and MHC protein on the APC. This specialized region that develops between a T cell and an APC is referred to as an immunologic synapse. So MHC proteins can be subdivided into two major groups, MHC class 1 and MHC class 2 molecules. So MHC class 1 molecule consists of one polypeptide chain encoded by an MHC allele known as a heavy chain associated with non-covalently with non-MHC polypeptide known as beta 2 microglobulin. So differences in the amino acid sequence of the heavy chain are responsible for dramatic changes in the shape of molecules peptide binding group. MHC class II molecules also consist of a heterodimer, but both subunits are encoded by MHC alleles. So both classes of MHC molecules as well as beta 2 microglobulin contain Ig-like domains and are thus members of the immunoglobulin superfamily. Whereas most cells of the body express MHC class I molecules on their surface, MHC class II molecules are expressed primarily by professional APCs. So the two classes of MHC molecules display antigens that originate from different sites within a cell, through some overlap can occur. So MHC class I molecules are predominantly responsible for displaying antigens that originate within the cytosol of the cell, that is, endogenous proteins. In contrast, MHC class II molecules primarily display fragments of exogenous antigens that are taken into a cell by phagocytosis. The proposed pathway by which these two classes of MHC molecule pick up their antigen fragments and display them on the plasma membrane are described here. Processing of class I MHC peptide complexes. Antigens located in the cytosol of an APC are degraded into short peptides by proteases that are part of the cell's proteasome. These proteases cleave cytosolic proteins into fragments approximately 8 to 10 residues long that are suitable for binding within a groove of an MHC class I molecule. The peptides are then transported across the membrane of the rough endoplasmic reticulum and into its lumen by a dimeric protein called TAP. Once in the ER lumen, the peptide can bind to a newly synthesized MHC class I molecule, which is an integral protein of an ER membrane. The MHC peptide complex moves through the biosynthetic pathway until it reaches the plasma membrane where the peptide is displayed. Then processing of class II MHC peptide complexes. MHC class II molecules are also synthesized as membrane proteins of RER, but they are joined non-covalently to a protein called Li, which blocks the peptide binding site of MHC molecule. So following the synthesis, the MHC class II 1I complex moves out of the ER along the biosynthetic pathway directed by targeting sequence located within the cytoplasmic domain of Li. So MHC class I and II molecules are thought to separate from one another in the trans-Golgi network, which is the primary sorting compartment along the biosynthetic pathway. An MHC class I peptide complex is directed toward the cell surface, where an MHC class II Li complex is directed into a transport vesicle that fuses with the lysosome. Where the Li protein is digested by acid proteases, leaving only a small fragment associated in the peptide binding group. So CLIP is subsequently exchanged with one of the peptide derived from the digestion of antigens that were taken into the cell by way of the endocytic pathway. The MHC class II peptide complex is then moved to plasma membrane where it is displayed. So once on the surface of an APC, MHC molecule directs the cell's interaction with different types of T cells. So cytotoxic T lymphocytes recognize their antigen in association with MHC class I molecules. They are said to be MHC class I restricted. Under normal circumstances, cells of the body can come into contact with CTLs, display fragments of their own normal proteins in association with their MHC class I molecule. Normal cells displaying normal protein fragments are ignored by the body's T cells because T cells capable of binding with high affinity to peptide derived from normal cellular proteins is eliminated as they develop in the thymus. In contrast, when a cell is infected, it displays fragments of viral proteins in association with its MHC class I molecule. These cells are recognized by CTLs bearing TCRs whose binding sites are complementary to the viral peptides, and the infected cell is destroyed. The presentation of a single foreign peptide in the surface of a cell is probably sufficient to invite an attack by a CTL. Because virtually all cells of the body express MHC class I molecules on their surface, CTLs can combat an infection regardless of the type of cell affected. So CTLs may also recognize and destroy cells that display abnormal mutated proteins on their surface, which could play a role in the elimination of potentially life-threatening tumor cells. Now, in contrast to CTLs, helper T cells recognize their antigen in association with MHC class II molecules. They are said to be MHC class II restricted. As a result, helper T cells are activated primarily by exogenous or extracellular antigen, such as those that occur as part of bacterial cell walls or bacterial toxins. So, MHC class II molecules are found predominantly on B cells, dendritic cells, and macrophages. These are the lymphoid cells that ingest foreign extracellular materials and prevent the fragments to helper T cells. So, helper T cells that are activated in this way can then stimulate B cells to produce soluble antibodies that bind to the exogenous antigen wherever it is located in the body. Then, distinguishing self from non-self. T cells gain their identity in the thymus. When a stem cell migrates from the bone marrow to the thymus, it lacks the cell surface protein that mediates T cell function. Most notably, it's TCR. So the stem cells proliferate in the thymus to generate a population of T cell progenitors. Each of the cells then undergoes the DNA rearrangement that enables it to produce a specific TCR. The cells are then subjected to a complex screening process in the thymus that selects 4 cells having potentially useful T cell receptors. So studies suggest that epithelial cells of the thymus produce small quantities of a great variety of proteins normally restricted to other tissues throughout the body. So production of these tissue-specific antigens is under the control of a special transcriptional regulator called AIRE that is present only in the thymus. According to this model, the thymus recreates an environment in which developing T cells can sample proteins containing a vast array of the body's own unique epitopes. So T cells whose TCRs have a high affinity for peptides derived from the body's own proteins are destroyed. The process of negative selection greatly reduces the likelihood that the immune system will attack its own tissues. So, humans lacking a functional AIRE gene suffer from an severe autoimmune disease called APECED, in which peptides derived from the body's own proteins are destroyed. So, this disease is called APECED, in which numerous organs come under immunologic attack. The generation of T cells require more than negative selection. When the TCR interacts with the foreign peptide on the surface of an APC, it must recognize both a peptide and the MHC molecule holding that peptide. So consequently, T cells whose TCRs do not recognize self-MHC molecules are of little value. The immune system screens out such cells by recruiting that T cells recognize self-MHC self-peptide complexes with low affinity. So T cells whose TCRs are unable to recognize self-MHC complexes die within the thymus due to a lack of growth signals, a process referred to as death by neglect. In contrast, T cells whose TCRs exhibit weak, low affinity recognition towards self-MHC complexes are stimulated to remain alive but are not activated. This process of selective survival is referred to as positive selection. It is estimated that less than 5% of thymic T cells survive these screening events. So, those cells that recognize class 1 MHC molecules are thought to develop into cytotoxic CD4, CD8 T lymphocytes, whereas those that recognize class 2 MHC molecules are thought to differentiate into helper CD4 plus CD8 minus T lymphocytes. Both types of T cells leave the thymus and circulate for extended periods through the blood and lymph. T cells at this stage are described as naive T cells because they have not yet encountered the specific antigen to which their TCR can bind. As they pass through the lymphoid tissues, naive T cells come into contact with various cells that either maintain their survival in a resting stage or trigger their activation. As they percolate through the lymph nodes and other tissues, T cells scan the surface of cells for the presence of an inappropriate peptide bound to an MHC molecule. So CD4 plus T cells are activated by a foreign peptide bound to a class 2 MHC molecule, whereas CD8 plus T cells are activated by foreign peptides bound to class 1 MHC molecule. So CD8 plus T cells also respond strongly to cells bearing non-self MHC molecules, such as the cells of the transplanted organ from a mismatched donor. In this later case, they initiate a widespread attack against the graft cells that can result in organ rejection. Under normal physiologic conditions, autoreactive lymphocytes capable of reacting to the body's own tissues, are prevented from becoming activated by a number of poorly understood mechanisms that operate outside of the thymus in the body's periphery. So, a breakdown of these mechanisms of peripheral tolerance leads to the production of autoantibodies and autoreactive T cells that can cause chronic tissue damage. Then, lymphocytes are activated by cell surface signals. Lymphocytes communicate with other cells through an array of cell surface proteins. As discussed above, T cell activation requires an interaction between the TCR of T cell and an MHC peptide complex on the surface of another cell. This interaction provides specificity, which ensures that only T cells that bind to the antigen are activated. So, T cell activation also requires a second signal called the co-stimulatory signal, which is delivered through a second type of receptor on the surface of a T cell. This receptor is distinct and spatially separate from the TCR. Unlike the TCR, the receptor that delivers the co-stimulatory signal is not specific for a particular antigen and does not require an MHC molecule to bind. The best study of these interactions occur between helper T cell and professional antigen presenting cells, that means dendritic cells and macrophages. Then activation of helper T cells by professional APCs. Th cells recognize antigen fragments on the surface of dendritic cells and macrophages that are loaded in the binding cleft of MHC class II molecules. A co-stimulatory signal is delivered to a Th cell as the result of an interaction between a protein known as CD28 on the surface of Th cell and a member of a B7 family of proteins on the surface of the APC. The B7 protein appears on the surface of the APC after the phagocyte ingests foreign antigen. If the Th cell does not receive the second signal from the APC rather than becoming activated, the Th cell either becomes non-responsive or energized or is stimulated to undergo apoptosis, deleted. Because professional APCs are the only cells capable of delivering the co-stimulatory signal, they are the only cells that can initiate a Th response. As a result, normal cells of the body that bear proteins capable of combining with the TCRs of a T cell cannot activate Th cells. Thus, the requirement by a Th cell for two activation signals protects normal blood body cells from autoimmune attack involving Th cells. Prior to its interaction with an APC, a Th cell can be described as a quiescent cell, that is, one that has withdrawn from the cell cycle. Once it receives the dual activation signals, a Th cell is stimulated to re-enter the G1 phase of the cell cycle and eventually to progress through S phase into mitosis. Thus, interaction of T cell within a specific antigen leads to the proliferation, that is a clonal expansion of those cells capable of responding to that antigen. In addition to triggering cell division, activation of a Th cell causes it to synthesize and secrete cytokines, most notably IL-2. Cytokines produced by activated Th cells act on other cells of the immune system, including B cells and macrophages, and also back on the Th cells that secrete the molecules. The source and function of various cytokines are very important. We have seen in this chapter how immune responses are stimulated by ligands that activate receptor signaling pathways. But many of these events are also influenced by inhibitory stimuli, so that the ultimate response by the cell is determined by a balance of positive and negative influences. For example, interaction between CD28 and a B7 protein delivers a positive signal to the T, leading to its activation. So, activation of a T cell leads to the trafficking of protein called CTLA4 from intracellular membrane to the cell surface. CTLA4 is similar to CD28 in structure and also interacts with B7 proteins of the APC. Unlike the CD28 B7 interaction, However, contact between CTLA4 and B7 leads to inhibition of a T cell response rather than activation. So, CTLA4 is thought to play a role in maintaining peripheral T cell tolerance. A need for balance between activation and inhibition is most evident in mice that have been genetically engineered to lack the gene coding CTLA4. These mice die as a result of massive overproliferation of T cell, as noted. So, insights into CTLA4 function has recently been put to clinical use. Next major topic is activation of B cell by Th cells. Th cells bind to B cells whose receptors recognize the same antigen. The antigen initially binds to the immunoglobulin BCR at the B cell surface. So, antigens taken up by B cells can be soluble proteins from the extracellular medium or proteins bound to the plasma membrane of other cells. In the later case, the B cell obtains the antigen by spreading itself over the outer surface of the target cell and collected the BCR antigen complex into a central cluster. The bound antigen is then taken into the B cell where it is processed enzymatically and its fragments are displaced in combination with MHC class II molecules. The B cell with the displayed antigenic peptides recruits a Th cell with the appropriate cognate TCR. So, recognition of the peptide fragment by the TCR leads to activation of the Th cell, which responds by activating the B cell. Activation of a B cell follows transmission of several signals from the Th cell to the B cell. Some of these signals are transmitted directly from one cell surface to the other through an interaction between complementary proteins such as CD40 and CD40 ligand. So, binding between CD40 and CD40 ligand generates signals that help move the B cell from the G0 resting state back into the cell cycle. Other signals are transmitted by cytokines released by the T cell into the immunologic synapse, separating it from nearly B cell. This process is not unlike the way neurotransmitters act across a neural synapse. Cytokines released by the T cell into the immunologic synapses include IL-4, IL-5, IL-6, and IL-10. So, interleukin 4 is thought to stimulate the B cell to switch from producing the IgM cell to producing the IgG or IgE class. Other cytokines induce the proliferation, differentiation, and secretory activities of B cells. Then, signal transduction pathways and lymphocyte activation. Hormones, growth factors, and other chemical messengers bind to receptors on the outer surface of target cells to initiate a process of signal transduction, which transmits informed into a cell's internal compartments. We saw in that chapter how a large variety of extracellular messenger molecules transmit information along a small number of shared signal transduction pathways. The stimulation of lymphocytes occurs by a similar mechanism and utilizes many of the same components employed by hormone and growth factor that act on other cell types. So, when a T cell is activated by a dendritic cell or B cell is activated by a Th cell, signals are transmitted from plasma membrane to cytoplasm by tyrosine kinase, similar to the signals described for insulin and growth factors. So, unlike the receptors for insulin and growth factors, lymphocyte antigen receptors lack an inherent tyrosine kinase activity. So, instead, ligand binding to antigen receptors leads to recruitment of cytoplasmic tyrosine kinase molecule to the inner surface of plasma membrane. This process may be facilitated by the movement of activated receptors into lipid rafts. Several different tyrosine kinases have been implicated in signal transduction during lymphocyte activation, including members of SRC and Treg families. SRC was the first tyrosine kinase to be identified and is the product of the first cancer-causing oncogene to be discovered. So, activation of the tyrosine kinase leads to a cascade of events and the activation of numerous signal transduction pathways, including activation of phospholipase A, which leads to the formation of IP3 and DAG. IP3 causes a marked elevation in the level of cytosolic CA2+, whereas DAG stimulates protein kinase A activity. So, activation of RAS, which leads to the activation of the MAP kinase cascade, activation of P13K, which catalyzes the formation of membrane-bound lipid messengers having diverse functions in cells. Then transmission of signals along these various pathways and others leads to the activation of a number of transcription factors. So NFK beta and NF80 and the resulting transcription of dozens of genes that are not expressed in the resting T cell or B cell. As noted above, one of the most important responses by an activated lymphocyte is the production and secretion of cytokines. Some of these cytokines may act as part of an autocrine circuit by binding to receptors on the cell that release them. So, like other extracellular signals, cytokines bind to receptors on the outer surface of target cells, generating cytoplasmic signals that act on various intracellular targets. Cytokines utilize a novel signal transduction pathway referred to as the JAK-STAT pathway, which operates without the involvement of second messengers. The JAK portion of the name is the acronym for Janus kinase, a family of cytoplasmic tyrosine kinase whose members become activated following the binding of a cytokine to cell membrane surface receptor. Janus is a two-faced Roman god who protected entrances and doorways. Entrances and doorways. Then STAT is the acronym for signal transducers and activators of transcription, a family of transcription factors that become activated when one of their tyrosine residues is phosphorylated by a JAK. Once phosphorylated, STAT molecule interact to form dimers that translocate from the cytoplasm to the nucleus, where they bind to specific DNA sequence such as an interferon stimulated response element ISRE. ISREs are found in the regulatory regions of a dozen or so genes that are activated when a cell is exposed to the cytokine interferon alpha, IFN alpha. As with the hormones and growth factors discussed, the specific response of a cell depends on the particular cytokine receptor engaged and the particular JAK and STAT that are present in that cell. For example, was noted earlier that IL-4 acts to induce Ig cell switching in B cell. This response follows the IL-4 induced phosphorylation of the transcription factor STAT6, which is present in the cytoplasm of activated B cell. Resistance to viral infection induced by interferons is mediated through the phosphorylation of STAT1. So, phosphorylation of other STAT may lead to the progression of the target cell through the cell cycle. Some autoimmune diseases associated with this: multiple sclerosis, type 1 diabetes, Graves' disease and thyroiditis, then rheumatoid arthritis, systemic lupus erythematosus, then inflammatory bowel diseases. Thank you for listening. So we just completed this portion. We'll be dealing with some other aspects later, not related to this topic, but some other topics related to immunology. Thank you for listening.